Evening, everyone. My name is Janerra Allen, and I am a PhD candidate at the University of Maryland, Baltimore County, where I am studying to get my PhD in electrical engineering. Today, I will be talking about my dissertation topic, which is on functional magnetic resonance imaging study of spatiotemporal brain dynamics in patients with schizophrenia. Thank you all for having me. So before we get started, I would just like to give a brief overview of my career timeline. So I attended the University of Wisconsin-Madison, where I majored in material science and engineering. While I was at UW-Madison, I did work in a neuroradiology lab that focused on stroke rehabilitation. And so that sort of was the foundation for my current work. In that lab, I had the opportunity to conduct neuropsychological testing, as well as I got to oversee some of the functional MRI monitoring. And we also had the rehabilitation component, which incorporated brain-computer interfaces to help those who were impacted by stroke to improve the mobility that was lost in their in their arms, so the upper extremity in their arms. And so with that different components, I knew that I wanted to further study functional MRI data, particularly looking at patients who are suffering from different neurological disorders. After I graduated from University of Wisconsin-Madison, I went on to work as a support engineer, and then I was ultimately promoted to support engineering manager at a telehealth company based in Hoboken, New Jersey, which is called Health Recovery Solutions. And then during my PhD, which I started in 2019, I was able to do internships at GE Research and Genentech. And this middle section here is where I'll be focusing the bulk of my work, which is, again, my title as stated here. I'm planning to graduate this May. And really the goal overall in the future is to continue to develop models that will not only improve analysis for disease diagnosis, but also for treatment. And this is speaking broadly to neurological disorders. So with that said, here's an overview of today's talk. I will give the project overview, background, and motivation, my approach, results, and impact. So just taking a step back, for this talk, I am looking into schizophrenia, which is considered a psychosis mental disorder. And in the US, it does affect 1 in 100 US adults. And so just to give a very quick high-level background, when we're thinking of neuroimaging methods, we're thinking of looking at the brain from a structural and a functional perspective. For this talk, I am focusing, on functional MRI here. And so functional MRI, if many of you are familiar with MRI, MRI is looking at tissue and organs of the body. The functional part of it is looking at the brain. I'll get more into that later, but we're trying to assess what is considered the relationship between blood flow and brain activity. And so the idea is that there is areas of increased blood flow, you would have increased brain activity. Thus, with decreased blood flow, you would have decreased activity. So that is what the focus of this talk is. And so on the right-hand side are some of the questions and approaches that we're trying to utilize. So the main overarching question is, what are the significant brain activation differences between those patients who have schizophrenia and then healthy normal controls? And with that said, we are using functional MRI as our tool so we can isolate the different regions of interest, or ROI, that show the greatest patient control differences within these groups. The gap in this research is suggesting that perhaps overall cognitive dysfunction is poorly understood because of the complexity of the data and trying to make sure that we are utilizing the most accurate neuroimaging method. And so we're trying to characterize this data. We're trying to figure out what type of methods we can use to best extract the most information. And the significance of this study is saying that if we can utilize both model and data-driven approaches, then we could look into how we can have targeted or localized treatments, because the idea is once you know some of the regions that are giving rise to these differences, then you can have treatment options that would help improve overall cognition. And one example of a localized or targeted treatment is the transcranial multi-site stimulation. So as I mentioned before, we are looking at functional MRI, this is saying that we are measuring the spontaneous bold signal over time. And so bold is the indirect measure of neuronal activity. I stated before that it is the relationship between blood flow, and saying that blood flow and brain activity are correlated. And so when we say spontaneous, you would imagine that the brain, as long as you are, you have activity there, you are awake, even when you're conscious or unconscious, there's always activity. And so when the scanner, whether you are at rest or doing a direct task, we're able to pick up on those signals. And so what we're highlighting here is that at different regions, they have their own corresponding time series signal, and time series is that bold signal for that given location. And so what we do with that information is that for a given study, we are looking at both the individual analyses, and I'll get to the next part, which is the group. But in the individual, we're trying to ask the question of, again, the spatial information and temporal information. So what is happening? When are these changes happening, and where are they happening? And so what you can get from these analyses is sort of view of different ways in which the brain is responding to stimuli, or the brain is at rest. And so you're trying to use this as a way to understand normal functioning, but also disrupted functioning, or decreased functioning, I should say. And so when we are able to collect all of the different subject information, what we do then is have the group analyses, where we can take a look at how can we best differentiate both groups, which we already know who's healthy and who has schizophrenia in this case, but we're trying to see if we can truly distinguish between the both. And what we have here is just a sort of chord, or this sort of diagram that is denoting that these are the relationships between the ROIs in the brain, and the red and blue are corresponding of blue is increased activity, red is decreased activity. And this is a particular example for one of the cases that we have. And so we'll get more into this later, but this is just an overview of how we go from subject level, individual subject level, to group level. So for this particular study, I mentioned that I go to UMBC, but for most of our medical data, we either collaborate with a medical school, or we get open source data from online. And so for this design, this is looking at resting state. Resting state, again, is looking at that spontaneous fluctuation. So your body is the body is just at rest. And so this was retrieved from the Maryland Psychiatric Research Center. And for this data set, we had approximately 63 healthy controls and 55, and we consider them on the schizophrenia spectrum disorder, which just means that they have different symptoms corresponding with their schizophrenia diagnosis. And one other thing that is very interesting is what we're trying to do, not only in having the neuroimaging data, but there's also that neuropsychological information too that we retrieve, which are some of the additional clinical measures that we take a look at, so that we can sort of show the relationships between the clinical measures and that imaging information, because the neuropsychological tests or the additional clinical measures are sort of the ratings that help the clinicians sort of assess where the patients are on these different scales. So you know that, you can recruit patients to your study, but you still need some level of baseline to understand, the severity of their schizophrenia symptoms. And so just to give an example, this brief psychiatric rating scale is just trying to assess their day-to-day, if they experience any levels of psychosis, but also we have this perceptual state and trait, which is asking questions related to their auditory. Are they hearing noises? Are they seeing things? And so when we think of schizophrenia, one of the primary symptoms are hallucinations. And so trying to see again how you can rate some of these patients is helpful when you're thinking of a treatment perspective. So the severity means a lot. And so when you couple that with the neuroimaging data, we try to get a more comprehensive view of what's happening with these different groups. So now that we have the overview of the study, I'll go into the approach. And so just take a step back. This is the full analysis for our data, but I'll just focus at the top. So the goal here is to understand, or how can we map brain dynamics, which is coming from this functional MRI data. And so, as I said before, we have all of the individual subject information coming from the fMRI scanner. And so this is part of our input. And so for this first part, which is CON, which is a toolbox, which is helping us to process the data, and what you would imagine, we have to account for any sort of errors or noise, people moving the scanner. So we're trying to just clean this part. And it's also helping us validate our analyses. But anywho, this first part is where we are trying to extract some of those connectivity measures. If you can recall, in a few slides before, there was that chord diagram, and this is where we would see what those region to region relationships look like. And so when we have this information coming from this first toolbox, what we now use this is as our new input coming into what we call the energy landscape analysis. And the energy landscape analysis is a tool in which we are now trying to assess, now that we have the ROIs, the regions that are showing the greatest patient control differences, we're trying to now assess what is happening at these regions. And so when we say what is happening, well, what are the levels of activation? Are we saying that, this is the areas or which, maybe there's decreased activation, and if it's decreased activation, is it based off of the energy of that region? And so this is just showing that it's based on the maximum entropy model and the Ising model, which are just physics-based models that are just trying to extract these energy measures. And so just to summarize that, basically, the first part is where we're acquiring the data so that it's pre-processed and validated, but we're using this as an input. So now we can look at what these brain activations are at these regions. So moving on to results and impact. So one of the questions we're asking, right? So we know that we're looking at the patient group and the control group. So the question here is, does the average connectivity, and this average because we're looking at it from a group perspective. So does the average connectivity differ between these two groups? And we have, a few hypotheses, right? Is it possible that the patient group would have higher connectivity than the controls? Is it possible that the inverse is true, that the controls would have higher connectivity to the patients? And as we can see, just not to cause any confusion, when I say red, I'm referring to these chord diagrams. And so this is showing that these are the ROI to ROI connections. So the connectivity between these ROIs may suggest that, there's either a positive connectivity or negative connectivity. And so we're trying to see which group is displaying the type of connectivity that would suggest that perhaps, schizophrenia patients may need treatment, right? And so with that, we wanted to go ahead and say that is there any indication that would suggest that patients have higher connectivity than the controls? And so because we are showing blue, so it's inverse, the case is that in fact, patients do not have higher connectivity in this particular data set. But there are some examples where you may see increased connectivity when you're looking at the different groups. But for this particular data set, we did not see that happen here. Yeah. And just again, when we're looking at these lines, they are showing the region to region connections, the relationships of that. and the clusters are formed based off of the anatomical properties and the relationships or the similarities of these regions and where they may belong to as far as a network. A network just meaning they may have similar characteristics if they belong to the auditory or the visual or default mode. So that's kind of what this is trying to approximate the relationship between the ROIs, but also how we would consider these clusters to form as well. And then again, just to wrap up, the connectivity maps are depicting the correlation between each ROI so the relationship of each ROI to every other ROI in the brain. And this is why you see some of these grayed out, is because those aren't significant. And here again, just to wrap up, this shows that patients in fact have a decrease because of that inverse relationship that we know to be true. And we sort of use this to validate, is it plausible biologically, which is saying that we would expect this to be true, which hopefully this is showing to be the case. And again, we saw that ring that had all those different connections, but one of the limitations with the energy landscape is trying to equally assess all those regions. So we have to figure out a way to reduce the number of regions. So we use Bartholini correction to reduce these down to the regions that show the greatest patient control differences. And so really, I'm just going to show that these are corresponding to these lobes and networks. And we use this as a guide so that we know for these particular groups, for these two groups showing the patient control differences, then these are the regions that are probably corresponding. So we want to say this is possible what is contributing to their hallucinations. So we may say that the visual, auditory, attention, sensory motor networks are involved in some of this processing when you're thinking of schizophrenia, but more so we're trying to now associate this with schizophrenia patients who suffer from hallucinations and primarily auditory hallucinations. So we want to show that relationship. And so we'll take a look at that in a little bit. So I mentioned before about the activation at these regions. And so energy landscape is approximating activation states based off of the number of ROIs. And so in this case, we had the ROIs number 8. And so we would have 2 to the power 8 number of activation states. We're trying to evaluate the energies or the frequencies of these particular states, just to see if this is corresponding to the activation and the frequency in which these states are visited in the brain. So now we're trying to show that we have these states. There could be a relationship of activation at these states, and being at this state, if you're there for longer, is it more stable? Does it mean that you have a better chance of not inferring information that is incorrect? So now we're just trying to understand or try to infer what is happening at the ROIs from the standpoint of knowing what the ROIs represent, but also knowing what these activation levels may represent. So we identified these states that we consider paired. And so we're trying to say that there is some stability there. And we're using this now as a threshold to see if these are something that we can now correlate with some of those neuropsychological testing methods that we mentioned earlier. And so, yes, we are testing the correlations to see what the relationship are between these particular states and those clinical variables. But before we get to that, this is a activity map of those 8 regions. And what we're showing here is the paired state, so that paired nature, and saying that these are the activities of these particular states. So we're saying that when a state is either in black or white, that they are active or inactive. And so the relationship of this is suggesting that these regions correspond to these particular networks, but also looking at the paired nature, this correlation or anti-correlation, that this may also be an attribute of the data, and saying that this may also give rise to some of that, that lower connectivity or disconnectivity that is happening. And so we want to use this to say that if these are the most significant states that show the greatest patient control differences, is this where we can sort of attribute this sort of levels of cognition and seeing if this is something that could be correlated with those clinical variables. And so with that, oh, and then this is just showing, sorry, I forgot to mention this part. This is just highlighting that if you look at the two paired from left and right, the leftmost of both of these paired have the same relationship except for the ones that are highlighted. So looking at this flip or this difference in I guess activity or inactivity, then we're trying to also suggest if there are of these 8, are there 4 that may also give the most reasoning for why this is occurring or why these are showing the most differences. So just for a quick discussion of those 8 regions, we know that these are showing the greatest patient control differences. We're saying that perhaps the abnormal energy landscapes between these regions may suggest that they're happening in these particular areas that are linked to processing. And well, these areas are related to processing vision and encoding visual memories, but also language processing and comprehension. And so when you would imagine when people are incorrectly taking in information or I guess referring to information, perhaps the relationship that they make in their minds, or if they're trying to recall to past experiences, there's something that is incorrectly happening, or there's relationships that are incorrectly forming. And so knowing that when we process information, how we envision certain things, this could also be linked to that. And so we're just trying to see if this is something that can be more concrete. And we're hoping if this is resting state, the next objective would be to look at task-based, which is more concentrated on certain tasks that would suggest that this could also relate back to this language processing and comprehension regions. So I did mention before that we were looking at the clinical measures between the patients and controls. And so one thing that was very, I guess, a bit disappointing was, yes, we identified the significant patient control differences from the imaging data, and that we inferred this using these different toolboxes. And there was a positive correlation when we looked at those particular states and the relation or the correlation with these variables, excuse me. But when you account for when you account for outliers and you account for trying to correct for skewness, I did some robust regression methods. I used Spearman rank correlation coefficient to try to correct for this, and I did some other to correct for skewness as well. And and what was again disappointing was no longer seeing that significance. So realizing that perhaps there are a few subjects that truly stand out, and so that would be something that we could probably look into in the future in terms of maybe that ties back into levels of severity. Maybe we can look at these different patient groups. And not as just, I guess, one patient group, but more so based off of their scores, or again, based off the severity of their schizophrenia. And same thing for maybe even for the controls. We could even do some type of assessment based off of if age, but in this case, age was not a strong factor in terms of looking at those patient control differences, but just trying to think of from a design perspective, if we could segment the data to look at it from different perspectives. But this is just highlighting quickly what these tests correspond to. And I didn't mention this before, but this is the brief psychiatric rating, the auditory perceptual, and the state and trait. And so just looking at these means, and you could see that this is all assessing more the negative symptoms. And so you would see here that patients tend to rank higher for those negative symptoms, which is probably corresponding to their schizophrenia symptoms overall. So really this work, as I said before, We're trying to identify the region-specific energy patterns that are relative to schizophrenia, and we're trying to continue to build a case as why functional MRI data is very powerful tool to identify hopefully connectivity biomarkers that are related to schizophrenia. And so we're trying to use this, and this is just an example of, I mentioned earlier, that TMS tool that is part of that targeted treatment. And so here you have these coils and trying to directly, or I guess not really directly, but trying to improve or I guess correct for that decreased connectivity. So this is more of I guess, an approach to see if this can be extended to schizophrenia, because currently this work is used to, I guess, improve depression or help with people who suffer from depression. So trying to see if this could be something that could also be extended to other areas. And so with that, I think I'm going to conclude. Oh yeah, this is just some of the work looking at schizophrenia. And so we just, again, we played with a few different things in terms of, you know, trying to assess if this is what we can continue to utilize. And again, I mentioned before, looking at resting state, but our goal is to also look at task-based, which is currently in the pipeline now. And then the end goal also is to see, well, this is more the theoretical part, but the next part is trying to see if our understanding of the brain broadly, but also from these neurological disorders, is how do you now understand how memory is stored? And so when you think about, for example, ChatGPT, we think about how data is inputted, how data is stored, how it can remember what you put, and how it can recall that information, and how it makes the associations for what's to come next. And so we think of this as a model that is very much brain inspired, but also how does the brain do these complex things as well, right? So knowing that if we look at it from this perspective of knowing how regions of interest play a role in terms of how we understand this complex functioning of the brain, but also trying to figure out how much does our knowledge, our experience play a role in how we recall it, how we recall, and how we are able to just function in our daily lives. So understanding how it builds and understanding short-term memory. So this is more so what we're trying to contribute to the field of just understanding how memory is stored and how memory is retrieved. So not only we can look at it from a diagnosis and treatment perspective, but also how do you also improve AI models? So kind of twofold in this case. And yeah, this is like my last slide, but next steps, my goal is to graduate, and I'll be working as a postdoc at APL. So yeah, that's all I have. Any questions? So should I stop sharing or?
Sure.
I don't know. Oh, if I stop sharing, and I could see the. Yeah.
I was just curious. So when you're doing the data gathering part for the resting state fMRI, how long are they actually at the scanner for?
Yeah, they're only a scanner for about a couple minutes. It does depend on the study design. For us, the subjects are only in the scanner for a few minutes, as I said, because it's only resting state. And so they are looking at we get both structural and functional information with that. And so when we're looking at the data, we're aligning the functional data to the structural data. But yeah, this is for a few minutes. Yeah.
And when you guys are developing these functional task-based paradigms, do you happen to know like which kind of paradigms they're considering using? Because I just imagine it's going to be different than a lot of clinical ones.
Can you repeat the question when you say?
Yeah, what kind of task-based paradigms are being considered for that part of it?
Yeah, so for example, in our task space, so the data that I'm looking at is assessing like sentence lists, word lists, reverse speech. So those are the types of different tasks, but tasks can vary. So we're not in control of the study design because we're not a medical school. So this is where the partnerships come in. And so we're trying to reason with, when we're looking at this data, what is going to help us with getting the most information pertaining to schizophrenia and in particular schizophrenia patients who are suffering from hallucinations. And so for that task-based data set, they are observing or they have the stimuli. Sometimes it could be provided to them visually, sometimes it's read to them. And so they're just responding to that to see. And now we're trying to look at, well, with these different groups, how are they able to respond to that particular stimuli? But for that task space, it is reverse speech, word list, sentence list. And I believe there's one more thing I may be blanking on, but that's for that particular task. But it can vary. It can, depending on the type, people can solve questions in the scanner, but it really just depends on for that particular group, what their goal is. And for this, because we're assessing, again, auditory hallucinations, we're trying to understand how the role of language processing is involved. So when you're looking at words and reverse speech, you're trying to, I guess, understand, this is the correct way, this is how it should look, and this other way is incorrect because it's reversed. So that's pretty much what that design looks like. Does that answer the question? Okay.
Yeah.
You looked at one patient like at multiple times to see how stable these patterns are, because obviously there's a big difference between somebody who's schizophrenia is well controlled at that time versus somebody who's having more.
That's
symptoms.
a really good question because unfortunately we don't look at progression. These are individual, these are just one-time points. It would be nice to know the different stages of their schizophrenia, how it may progress, the type of medications they're on. Some of this stuff is de-identified, and so we don't have a lot of control over, basically not only the design itself, but also, the type of demographic information we would like to have. So in this particular case, we have information on sex, smoker and non-smoker, age, and of course the imaging scans. But it would be nice again to sort of track over time. One thing that stood out, but I think because this is more of, this is not focused on rehabilitation. So for example, to take a step back, in undergrad, they had different time points because you were bringing in people who were affected by stroke. And the goal of that was to help improve their, upper extremity. And they actually had the component to try to get them to regain the upper extremity. So you would expect to see different time points to see if they would improve. But for this, this is just a singular time point. Yeah. Yeah. But yeah, in a future study, but I think also because the goal isn't treatment quite yet, we're trying to make a case for why different types of treatment would be ideal. So if they're currently taking medication to stabilize the symptoms, we're trying to say that if you can find those regions that are showing that, okay, this is where the connectivity is negative or is worse off compared to controls, then what other types of treatment can we assess so that we can target those particular regions? Because medication isn't going to just target, those areas. So that's kind of what we're trying to make the case for. But yeah.
Another question. So how small are these ROIs that you're trying to correlate to, come down to just Solvit or a gyrus?
When you say small, what do you mean by small?
Yeah, I guess what's the resolution of these buckets that you're trying to
Oh, that's a good question. I don't think I recall off the top of my head. I just know,
It's like a list as like broad areas.
yeah,
Yes.
yeah, I will have to, I don't want to give you a wrong estimate, but yeah, I'll probably have to.
So I guess my other question with that is how are the activation maps assigned to the anatomical maps? Is it an automatic thing? Because I guess not everyone's functional anatomy is the same.
Absolutely.
Somebody's got a tumor or something like that.
Yeah.
Their language center can be on the other side.
That's
So how is that being determined? Is it just completely automated, or do you have some human oversight going in, looking in and trying to decide where,
Mhm.
you know, these pathways are?
That's a good question. So, yes, you're right. We do try to, correct for, we do try to account for different, skull sizes, brain sizes. And so that's where we try to normalize the data. So we do normalize to a certain brain template, that was averaged over, I guess, a couple hundred, couple thousand people. And based off of that average, then we align that to this particular atlas. There are different types of atlases out there, templates out there that exist. The one that we're using is called the Brain Nectome Atlas, which is looking at 246 ROIs or nodes in the brain that correspond to those lobes. But we do align all these brains so that it's normalized to a singular template that is based on, I think, the Montreal neurological template. But yeah. So using both for the normalization, but also trying to attribute those ROIs and aligning those ROIs to the time series is how we get that both spatial and temporal information.
It's like a difficult problem to solve. I guess that's a source of noise. It's
Mhm.
for different atlas.
Yeah.
But
Yeah. And it's making those assumptions too that, here we're using this normalized template. Like you said, you have people who and I think that's also where the criteria comes in, and, you may not want people who have tumors. You may not want people who, have other psychosis related disorders. So just trying to, I guess, refine it from, as much of a standpoint as you can from the beginning. And that's where, some of the pre-processing and the noising steps come in. But yeah, do our best to account for that. Yeah. Oh, thank you.
Make sure the chat doesn't have questions.
Oh, yeah. Okay.
So this isn't directly on your talk, so you might say, I don't know, Lisa.
Oh, okay.
But can you talk about the TMS thing? Because they like advertise it on the radio now. And I'm
Really?
like, yeah, TMS treatment centers, and I'm
I
like, it
didn't know
sounds
that.
creepy. Does it actually build connections? Like, what is the?
So it doesn't build connection. So I'm gonna speak very briefly because that's not my area of expertise. But there is this magnetic coil, and they're trying to say that they're sending like this like stimulation to the brain, and it's supposed to be targeted in a way that I don't know if it's supposed to be customized to every individual, but you would imagine that the strength of it would hopefully correspond to, like, if this is happening in the brain, you could probably focus it on where it's happening in the brain and then send that stimulation there. And so, yeah, that's the overall concept of it, is to, have that to be a way to help strengthen those connections that way through that stimulation, magnetic stimulation, those coils. Yeah. I mean, I haven't seen it in practice, but in theory, it sounds like it would be hopefully effective. Yeah. So.
I have a question. Oh, yeah.
Oh, okay.
Are you gonna be continuing this work there, or?
That's a good question. So I just heard back, and I am expected to come up with my own proposal. And so ideally, I wouldn't continue my advisor's work. I would have to start from scratch, and it has to be within, certain constraints, right? Because it's dependent on, where the funding is. It's dependent on, the population. The interesting thing is that I guess with APL, I don't know if this is truly public, but they do work with the government. And so you would imagine that there are different populations. So you have the civilian population, you may have veteran population, you may have active, warfighter population. So keeping that in mind, I don't quite know, who I would want to focus on. But again, it depends on, where most of the funding is coming from. What are some of the, I guess, most dire need when you're thinking about what's happening in the world. But I would want to definitely stay within the neurological space. The role is health and human machine. So they look at all types of different assessment data. So it could be cardiovascular, it could be neurological. And then also the other thing is, when you have these multimodal approaches, how do you assess all these, using different scales? So I don't know if that was something that I would want to jump into right away. But yeah, hopefully with some guidance, I'll have more information on the type of project I could possibly study. So.
Looking forward to my APL tour.
So.
Questions? Thank you.
Thank you.
Thank you everyone online for being with us. We'll see you again soon.